All right. Hi, everybody. I'm Madeline with Lingso Garden Materials, and thanks for joining our clinic today. Today, we're joined by Nick Landolfi from our local Master Gardeners, and he's going to be teaching this class, Seeds, Exploring Anatomy and the Art of Preservation. Just one quick note, all questions will be addressed at the end of class. Due to some recent security issues, the chat is not open, but any messages that you send will be received by myself and Nick. So we'll cover all of those after Nick finishes his lecture. And Nick, thank you so much for joining us. The floor is yours.
Thank you, Maddie. As Maddie indicated, today I would like to talk about exploring seed anatomy and the art of seed preservation. Everyone knows what a seed is, but it is easy to trivialize their capability and potential. Here is a comprehensive definition of a seed. A fully developed, fertilized ovule of a flowering plant. An ovule is a plant's female germ cell, essentially an egg. A seed includes an embryo, a nutrient reserve, and is encased in a protective outer layer known as the seed coat. Seeds serve as the main means by which plants reproduce and spread to generate new plants. It is believed that humans appreciated the value of seeds over 12,000 years ago and began saving seeds for three main reasons. Number one, to assure a reliable food source. Foraging and hunting for food was tedious and unpredictable. Being able to plant seeds and harvest in a specific location was the opposite. Number two, they wanted to maintain desirable traits. Historians believe that one of the first characteristics that early seed savers wanted to focus on was the non-shattering trait. Plants evolved to maximize seed dispersal, an evolutionary advantage. And in wild grains, the seed heads shatter and drop from the plant as soon as the seed matures, spreading the seeds on the surrounding ground. This made collecting large volumes of grain difficult. Timing had to be precise. You didn't want to pick up individual grain from the ground. By saving seeds from plants that didn't shatter, early seed savers developed strains that were much easier to harvest. And finally, they were unknowingly optimizing their plants to local conditions. The plants that grew the best in that environment produced the most seeds and thus were selected for the next planting, optimizing the saved strains to the local environment. This is obvious, but I think the magnitude of a seed's potential is rarely appreciated. A 1/8 inch tomato seed, provided with sunlight, air, water, and nutrition from the soil, can grow into a fully developed plant, leaves, stems, roots, and fruit, that is several orders of magnitude larger than the side of the seed. And all this can take place in 3 to 4 months. Another example, a 3/4 inch acorn under those same conditions can develop into a tree 800,000 times heavier and 3.7 million times the volume of the original acorn. Of course, in this case, it takes decades. Seeds come in a variety of shapes, even though they all have the same function, which is to propagate the next generation of the plant. Here are some examples. I'm sure you can identify some by sight, but others may stump you. The shape of a seed provides a clue as to how they are dispersed by the plant. It is an evolutionary advantage for a given plant to spread its seed across a large area. Some seeds are packaged in such a way to enhance their spreading. Here are some examples of seeds for which wind is the primary method of dispersal. Including the wind, there are 4 basic mechanisms plants use to disperse seeds, as shown in this illustration. Seeds easily dispersed by wind, examples of which I have just shown you, have a structure that can catch the wind, allowing the seeds to move away from the parent plant. Some seed pods physically expel their seeds. Here are a few examples, but you may be more familiar with the California poppy, which literally pops its seeds from the seed pod. Seeds encased within a buoyant structure can be spread by floating on water. Examples are water lily and coconuts. Animals play a huge role in dispersing seeds, and animals include insects, birds, reptiles, mammals, including humans. This can occur by consumption and seed release in the feces. For example, a bird eating a fruit, the seeds ending up in their droppings. Physical attachment to the animal, for example, a thistle. Or actually actively moving a seed. If you have squirrels in a local oak tree, I am sure you have observed oak tree seedlings in your flower bed, vegetable gardens, and even potted plants. And of course, you planting seeds wherever you desire is another method of dispersal. We hear a lot about artificial intelligence these days, but a seed is an exquisite example of natural intelligence. Seeds are sensors. They can monitor a variety of environmental parameters. These include moisture, temperature, light, oxygen, nutrients, and hormones. Let me give you a little detail on each of these. Seeds monitor the moisture content of the environment. Water uptake or imbibition by a seed triggers metabolic activity and initiates the germination enzyme production. The presence of water is mandatory for germination. Seeds are temperature sensitive. The membranes and enzyme in a seed are influenced by specific temperature ranges and only become active within that range. In addition, some seeds, before they will germinate, require cold stratification, which is exposure to low temperature and moist conditions meant to indicate winter. Seeds can detect light levels. They contain molecules called phytochromes that are sensitive to light and indicate to a seed whether it is on or below the surface of the soil. These phytochromes can be inhibitory or stimulatory to germination. Some seeds, usually smaller seeds, are positive photoblastic, meaning they will germinate best when exposed to light. Examples are lettuce and celery. Others are negative photoblastic and germinate best in darkness. Examples are onion and spinach. Many seeds are light neutral and germinate equally well in light or dark conditions, such as tomato. Seeds can sense the oxygen level of the environment. Diffusion of oxygen into the seed is mandatory for the required respiratory activities for germination. A seed can detect the presence of nitrates, a key nutrient source for plants. The presence of nitrate is a general indication of the fertility of the soil. And finally, a seed can internally monitor the levels of two hormones, abscisic acid and gibberellins, that maintain dormancy or stimulate germination respectively. As the ratio of these hormones changes, germination is initiated. Now let's talk about seed anatomy. This diagram, or one similar, you may remember from your elementary school biology. This depicts the structure of the two main types of seeds, monocotyledon, or monocot, and dicotyledon, or dicot, represented here by a corn and bean seed respectively. Let's focus on their similarities. Both have a tough seed coat, also called a testa. This is a durable structure that protects the seed from damage, dehydration, and pathogens, but is also central to the ability of the seed to act as a sensor. Every parameter the seed senses must pass through the seed coat. The seed contains an embryo, which has developed the fertilized ovule and contains everything needed to develop into a mature plant. The cotyledon are the leaves of the embryo and provide nourishment to the embryo as it develops. This is where monocots and dicots differ, in that monocots have a single cotyledon, while dicots have two. There are many other differences between monocots and dicots, but since we are focused on seeds today, we will leave it at that. Finally, within the seed is the endosperm, which is the food storage area for the embryo. This contains starch, oils, and protein, which provide initial nourishment for the embryo during germination. Now, there's much more detail to the anatomy of a seed. In reality, the seed coat has 2 layers. The embryo can be divided into multiple substructures, but I don't think that detail and terminology is pertinent to this discussion. It is sufficient for you to understand that a healthy seed contains 3 main components: a seed coat, food storage, the endosperm, and a viable embryo. Now, this figure is mostly accurate, but it does have a significant omission, which was also absent from your elementary school lesson, because it is a relatively recent discovery. And that is a microbiome. All seeds contain a microbiome. You're most likely aware of the definition of a microbiome. It is the collection of all the microorganisms, including bacteria, fungi, viruses, protozoa, and archaea, that live on or within an organism. Virtually every multicellular organism has a microbiome, and it is essential for the health and survival of that organism. I am sure you know we have a microbiome, and the microbiome associated with our digestive tract is the focus of the currently $87 billion probiotic industry. Now, the discovery of seed microbiome is relatively recent. Until the late 1800s, the interior of a seed was presumed to be sterile, that is, microorganism-free. It is really only in the last 60 years that botanists have accepted that seed-associated bacteria truly exist, as that is when bacteria could be isolated and grown from seeds. Only in the last 10 years has the concept of a functional seed microbiome come to be appreciated. Of course, plants have a microbiome, and it can be broken down into 2 varieties: that associated with the phyllosphere, or the above-ground portion of the plant, the leaves, stems, flowers, and fruit, and that associated with the rhizosphere, or the underground part of the plant, the roots and the surrounding area. As with all microbiomes, it is essential for the health of the plant. It breaks down organic material to release nutrients. It assists the plant in absorbing those nutrients, and it can compete with or block plant pathogens. The seed microbiome arises from 2 sources. There are contributions from both the parent plant and the environment. These contributions can include organisms that live on the outside of the seed, referred to as ectophytes, and organisms that are actually contained within the seed, referred to as endophytes. With the understanding that seeds contain microbiomes, seed scientists came to the realization that seed microbiome manipulation could be beneficial. They recognized that they could impart to the seed microorganisms that could have a direct effect on the seed, such as enhancing nutrient acquisition, phytohormone regulation, germination, and root development. In addition, microbiome manipulation could have an indirect effect on the seeds in terms of pathogen control, initiation of systemic resistance, and alleviation of environmental stress. Here is an example of the potential of seed microbiome manipulation. Union Biosciences is a plant biotechnology firm in England that has a suite of products that serve the agricultural industry. One of the areas in which they are active is seed microbiome manipulation. This slide shows the results of one such manipulation. On the left is an untreated seed. On the right is a seed that has been treated with a proprietary mixture of endophytes. You can see the astonishing difference post-germination. While the untreated seed has visible hairs, they are dwarfed by those on the treated seed's roots. In addition, the treated seed has twice the number of lateral roots as does the untreated. Unium has data indicating that this translates into an increase in both above and below ground biomass, as well as crop yield. As you can see, seed microbiome manipulation has the potential to enhance multiple seed characteristics, including optimizing nutrient uptake, protection from competition, improving the quality of soil and water, stimulating metabolism, and increasing tolerance to abiotic stress. That pretty much concludes what I would like to say about seed anatomy. I hope you now have a good grasp of the parts of the seed and the functions they perform. Now, let's talk about collecting seeds and saving seeds for the future. Properly executed, this can provide an inexpensive and satisfying strategy to deliver seedlings for your garden. However, you must have a plan. There are a few things you need to consider. Number one, know your source plant. Not all plants are suitable for collecting seeds, so you want to make sure the plant you are focusing on meets certain criteria. Number two, have a collection strategy. Number three, allow the fruit or vegetable to mature completely before collection. This will ensure that the seeds are fully developed and germination competent. And finally, process and store the seeds correctly to maximize future germination. I would like to elaborate on each of these. Know your source plant. It is important to know the lineage of the plant of interest. There are really three types of plants gardeners deal with: open pollinated, heirloom, and hybrid. However, only two of these are worth saving seeds from. Open pollinated and heirloom are very similar. Both are naturally pollinated by wind, insects, or birds. Both will produce seeds that are true to type. This means that the seeds will produce plants very similar to the parent. They are genetically stable over generations. They differ in that heirlooms have a long history, being passed down through generations and selected for taste, culture, or tradition, and not necessarily yield. Definitions of an heirloom variety differ. Some consider pre-World War II varieties as heirloom, while others specify a history of 50 years. Heirlooms are a subset of open pollinated, so all heirlooms are open pollinated, but not all open pollinated are heirlooms. Both are suitable to save seeds from. Examples of popular heirloom tomatoes, which are also open pollinated, include Cherokee Purple, Brandywine, and San Marzano. Kentucky Wonder is a popular heirloom bean variety. Hybrid plants are very different. They are produced by crossing two distinct varieties and are hand-pollinated by the seed company. This produces a seed that will yield a hybrid plant that possesses characteristics of both parent plants. Hybrid plants are also referred to as F1, shorthand for first filial generation. Popular hybrid or F1 tomato varieties include Sun Gold, Better Boy, and Celebrity. Often the parental partners of hybrids are kept secret. The Sun Gold hybrid was developed by the Tokita Seed Company in Japan in 1992, and the parent varieties are still a corporate secret, although Brandywine has been speculated as one of the parent strains. These hybrid plants do not breed true. That is, they will produce seeds that will develop into plants with differing characteristics. I want you to have a good understanding as to why you should not save seeds from hybrid plants. So let's go through some very basic genetics. This is how hybrid plants are created. Two plant varieties with different characteristics are selected to hybridize. In this simple example, one variety has sweet but small fruit, while the other bears large but bland fruit. The two plants are grown separately, most commonly in greenhouses. Pollen is collected from one plant, while the anthers, the male part of the flower that contain pollen, are removed from the other plant. The collected pollen is applied to the stigma, or female part, of the plant from which the anthers have been removed. The fruit from that hand pollination will contain seeds that will grow into hybrid or F1 plants that have characteristics of both parents. In this example, large, sweet fruit. The seed company collects and packages these, and they are the hybrid or F1 seeds that you purchase to plant in your garden. The problem occurs if you save seeds from the fruit of a hybrid plant, and here is why. You purchase your hybrid seeds to plant them. The plant grows, and in the case of Sungold, produces copious amounts of sweet cherry tomatoes. You really like Sungold tomatoes, so you decide to save seeds from your plants. This is a mistake, and this is why. Unlike open pollinated and heirloom varieties that breed true, each of the seeds saved from a hybrid plant will be different. The seeds will produce plants and will most likely produce tomatoes, but each plant's characteristics will be different. In this simple example, some plants will produce sweet, large fruit, but some will produce bland, large fruit, sweet, small fruit, or bland, small fruit. So while there is a chance you may grow something similar to a Sungold, it is much more likely you will get something very different. This is why you should not save seeds from a hybrid variety. You should purchase them as needed. Devising a collection strategy is the second consideration for saving seeds. Know the plants from which you are interested in saving seeds from the time that they are planted. Now, if you're only growing a single plant, this isn't a concern. It is the only plant from which you will have seeds to save. But I always grow 2 or more plants of a variety in which I have interest. This way I can watch the plants develop and can target saving seeds from that which is most vigorous. I also pay attention to how early the plant bears fruit and the quality and size of that produce. Ideally, you want to save seeds from a vigorous plant that produces abundant, high-quality produce. The fruit or vegetable from which you want to save seeds will almost always be past the point at which you would harvest for consumption. Ideally, you want the produce to completely mature on the plant. Here are some examples. In each case, the fruit or vegetable that I would harvest for consumption appears on the left, and that I would use to save seeds appears on the right. The jalapeno on the left will certainly have seeds, but they might not be completely mature. To save bean seeds, allow them to completely dry out on the plant. And while an 8-inch zucchini may have mature seeds, your chance of success is much greater from a version allowed to grow much larger. I think this is obvious, but I will mention it. You want to select a suitably normal fruit or vegetable for seed saving. Physical defects may be passed on. Now let's talk about how to save seeds. Seeds can be classified into one of 2 categories based on how they are encased within the fruit or vegetable. These categories are dry and wet. Dry seeds are created by plants whose produce dry out upon maturity. There is no pulp around the seeds, and frequently the seeds are dispersed when the fruit dries out. Examples are bean, peas, grains, and sunflowers. Wet seeds, as the name implies, are produced by plants whose seeds are embedded in moist pulp or gel. These seeds need to be cleaned before they are dried and saved. In many cases, all that needs to be done is rinse the seed a few times in water to remove any attached pulp. For example, cantaloupe seeds. Some seeds, such as tomatoes and cucumbers, require more treatment. While some wet seeds are easily washed and separated from pulp, such as melon seeds, it can be more difficult to remove the material in which tomato seeds are embedded. The pulp or gelatinous coating around tomato seeds is a germination inhibitor that suppresses germination inside the fruit. In nature, this gel will break down when the fruit decays and the seeds become exposed to the elements, or will be removed in the digestive system of an animal that eats the fruit and then excretes the seeds. It is desirable to completely remove this material prior to seed storage. A simple fermentation technique can be used to remove the gel surrounding the seeds. Remove the seeds from the tomato and place them in a small amount of water in a container that is open to the air. Allow this to incubate for a few days, or 5 to 7 days at room temperature. After a few days, you will see mold growing on the surface of the water. This is the product of mold spores in the air, which have landed on the water surface. Allow the fermentation by the mold to continue for 5 to 7 days. This will digest the materials surrounding the seeds. Afterwards, fill the container with water and pour off anything above the sunken seeds. Do this until the water is clear. This will remove any debris and non-viable seeds, which will float. Place the wash seeds on a napkin or paper towel to dry. Separate the wet seeds or they will dry into clumps. The seeds will stick to the paper but can be easily scraped off. Alternatively, placing the seeds on a coffee filter to dry will prevent sticking. Make sure to clearly label your seeds at all steps to avoid confusion. While tomato and bean seed collection are straightforward, collecting some seeds is more challenging. For example, onion seeds reside in the flower or umbel at the top of the onion stalk. Harvest the umbel when it begins to dry and invert it over a container. The seeds will naturally drop from the umbel as it completely dries out, allowing you to harvest and store them. You know that if you want to harvest basil throughout the summer, you need to continually trim back the plants to prevent flowering. However, if you want to collect basil seeds, you need the plants to flower. And believe me, the bees will thank you for that. Trim off the flowers as they begin to turn brown, then use a strategy similar to that for onion seeds. Invert the flowers over a container and allow to dry. The seeds will fall from the flowers and can be collected and stored. Once you have collected your seeds, you need to devise a storage system. There are many commercial options. A search on Amazon for seed storage will yield over 800 results. And there are many storage systems not specific for seeds that could be applicable. This is what I use. I store individual seed types in these glassine envelopes, and then store these along with commercial packets and the snap top food storage containers. For larger quantities of seeds, I use herb jars. I put a few silica gel packets in each container to absorb any moisture. And I can't stress enough how important it is to label everything. You want a relatively stable environment in which to store your seeds. Someplace dry, dark, and cool is preferred. I use a shelf in my garage. A refrigerator would be ideal, but there are frequently space considerations. Properly collected and stored, most seeds are good for at least 2 years, with some remaining germination competent for 5+ years. Here is a seed viability chart from Homestead and Chill. I'll let you look this over for a few seconds. There are documented examples of seeds remaining viable for extraordinary periods of time. A 2,000-year-old Judean date palm seed recovered during excavation at Herod the Great's palace on Masada in Israel germinated in 2005. This tree had been considered extinct since about 500 CE, or AD if you prefer that nomenclature. This is a picture taken about 15 years post-germination. The second oldest documented seed germination is a 1,300-year-old sacred lotus seed recovered from a dry lake bed in China. You can assess the germination rate of your seeds at any time by doing a simple germination test. Place 8 to 10 seeds on a damp paper towel. Fold over the paper towel to cover the seed. Place in a sealed plastic bag to prevent drying and keep in the dark for 5 to 7 days. Viable seeds will sprout, giving you an estimation of the germination rate. In this example, the germination rates were 90%, 100%, and 80% for the pepper, melon, and tomato, respectively. I hope it is obvious that seeds are an essential resource. They are the foundation of global food security, ecosystem sustainability, and biodiversity. They are crucial for food production. They are the starting point for all agriculture. Diversity. Seeds contain the genetic material that specifies size, flavor, disease resistance, and drought tolerance. Economic value. The world's agriculture industry is estimated to be $14 trillion, and it is all based on seeds. Environmental resilience. Seeds adapted to local conditions can minimize the need for fertilizers and pesticides. And finally, cultural value. Many cultures preserve seeds important to their history and cuisine. Thankfully, many appreciate the incredible value and potential of seeds, and there are over 1,750 seed banks across the world to safeguard biodiversity, provide insurance against crop failures, support future research and plant breeding, and protect against corporate seed control. There are international seed banks, native and regional seed banks focused on specific crops or wild plant species. But in closing, there is one seed bank I would like to tell you about. This is the seed bank in Svalbard, Norway, and is literally at the top of the world. Svalbard is a Norwegian archipelago between mainland Norway and the North Pole. One of the most northernmost inhabited areas, Svalbard is known for its rugged remote terrain of glaciers and frozen tundra. Summer brings the midnight sun, sunlight 24 hours a day, and this is the location of the global seed vault. The Svalbard Global Seed Vault is operated by the Crop Trust, an international nonprofit dedicated to conserving crop diversity and making it available for use globally, forever. It opened in 2008 and currently has 1.3 million seed varieties in storage, which is about 1/3 of its capacity. It is kept at 0 degrees Fahrenheit, about 18 degrees centigrade. What you see here is simply the front door of the facility. This is a schematic of the entire seed bank. You can see it is embedded into the mountainside, giving it extraordinary physical protection. Its location in such a frigid environment was selected because of the year-round low temperature. But global warming has raised concerns on that assumption. You can also see it is comprised of 3 individual seed chambers. For reasons unclear to me, they started storing samples in Chamber 2, which is now at capacity. Current samples are going into Chamber 1, and Chamber 3 is awaiting the collection's expansion. In conclusion, I would like to reemphasize that the remarkable seed has incredible capacity and potential. Seeds contain the complete genetic code for a new plant, can remain dormant for years and even centuries, can transform dramatically from dormancy to active living in a very short time. They evolve and adapt. They come in a variety of shapes and sizes, and they feed the world. I would like to thank Maddie for moderating this discussion, Terry Lingso and Lingso Materials for supporting this seminar series, and all of you for your attention today. I can now attempt any questions you have. Maddie, are there questions?
All right, well, let's give everybody a chance to send some questions in. Again, if you message in the main chat, they'll come straight to me and Nick and we can moderate from there. I do have a question for you, Nick, because I have been myself experimenting with seed collection and I got married this year, so I grew poppies for that. I'm trying to harvest the California poppy seeds from my wedding and as you mentioned at the beginning of the presentation, they tend to explode once they're dried out. So I've been able to collect a few and I've had plenty of seeds that unfortunately are lost during the process. I still have plenty of green seed pods on those plants though. Is this a sort of plant where I can harvest it while it's green and let it dry naturally inside?
Yeah, my strategy for poppies is to let sort of 1 or 2 seed pods on the plant pop and then collect the remainders because that means they're close. And then I'll put them in a jar and simply let them pop away. And then it's very easy to remove the seed pods and the seeds all fall at the bottom.
All right, I figured that was probably the answer. I've got a whole bunch of green pods that I harvested and I've just kind of been keeping an eye on them. But yeah, really neat information. All right, and I'll give it just a minute. I don't have any questions yet. Oh, here we go. Right, this is Sherry and she's just thanking you. You gave the best explanation she's ever heard as to why you shouldn't harvest a seed from a hybrid plant. And I agree, that was great information. I've always wondered myself.
Well, thank you. That was, I know it's kind of tedious to listen to and go through, but I think if everyone has a good understanding of why, it'll save you a lot of time in the future.
Yeah, and a lot of frustration too. We do have somebody with some more interest in the native plant harvesting. Do you have any tips or tricks for our California natives? Is it much different than the vegetable harvesting?
You know, I don't have
The herbs?
I don't have specific experience in that area. My feeling is that because they are native, basically letting them go to seed where they are will result in the plants reseeding. Now, if you want to collect seeds to put them elsewhere, then it's just a matter of timing. You know, watch the flowers until they turn into some type of seed pod as they dry out, and wait until you can physically detect seeds in the flower, and then save those. I know in my backyard there are areas where I have some flowers that come up, and I simply wait until the seed head is dry, cut it off, and then just basically toss it in the same area with the hopes that that will degrade and the seeds will be dispersed in the area and then will germinate in the next season.
Yeah. And most seed pods have from dozens to hundreds of seeds inside of them. So you have a pretty good germination rate on those, I expect.
Yes.
Yeah. All right. And then let's see. We have a question from Peter. They've seen F2 labeled seeds. Is there something better that people should be purchasing? if they want to avoid getting the F2 and they want to be able to harvest their own seeds in the future.
So a hybrid plant is also an F1. And so the F1 seeds will result in a hybrid plant. Any seeds collected from a hybrid plant are referred to as F2, the familial second generation. And these are the ones that will have the incredible variability. You know, the example I gave you was very simple, basically 2 traits. But in reality, there are literally thousands of traits that a plant has. And so harvesting F2 seeds will give you a tremendous variety of characteristics in the plant. So it's not, yeah, I know of no cases where F2 seeds are available for sale. And so you want to limit yourself to F1 seeds. And unfortunately, you need to just, if you really like that variety, you need to just buy that variety whenever you need them. I know I like Sungold very much. I like Celebrity is also a hybrid tomato. And I just basically buy the seeds every couple of years. They're usually good for at least 3 years for germination. And so you just have to be aware of that.
All right. And here's on the subject of tomatoes, I have somebody who earlier in your lecture, you mentioned there's a substance on the inside of the tomatoes that will keep the seeds from sprouting. However, she's actually found some tomatoes when you slice them open, there's seeds that have sprouted inside of the fruit. Why
Yes.
does that happen? And are those plants viable to replant?
Yeah, I don't know why that happens. It's clearly a failure of the inhibitory activity of the gel. That's why the gel exists, because if you think about it, the inside of a tomato is a very receptive environment for germination. It's moist and very nutrient rich. So I think on occasion you will see that, but the true reason for that gelatination material is to basically prevent that very thing.
All right. And then we have a question about Cosmos. Sometimes when they deadhead the flower before it's completely dried, or sometimes they deadhead before it's completely dried. And the question is, if they dry it out in a paper bag, will it be viable or will it be less likely to germinate if it's harvested early?
I think there's a high probability it will be viable. You know, deadheading a flower, cutting it off, you don't remove immediately all of the nutrition that is going to finish the seed's development. There's still some that's in the flower, and over time, that nutrition, so to speak, will be delivered to the seed and allow it to mature. So as long as you don't deadhead too early, as long as you let the seed generation progress at least a bit, there's probably a good chance you'll get fertile seeds from that strategy.
Okay. And then we have 2 questions on refrigeration of seeds. So are there some seeds that you should keep in the refrigerator or freezer before planting?
There are some seeds, well, the seeds that require cold stratification, if your environment is not sufficient for that, that would be worthwhile. I know Japanese maple seeds, for example, I would put those in my freezer for a couple months in the winter before I would try to germinate those. again, to replicate basically a cold winter. But other than that, the reason you store seeds, or the reason refrigerated environment is ideal for seed storage, is basically you're just slowing down any metabolism that's going on in the seeds. So you have the, if you have seeds that you keep refrigerated, the chances are very good that they'll show a higher germination rate for a longer period of time than seeds you just have out on your counter or out the door, because the degradation that naturally occurs over time occurs much slower at lower temperatures.
All right. And can you please repeat the website which gives seed life?
That is called Homestead and Chill. They put out a weekly newsletter. And they usually have 2 or 3 articles. And this particular table came from an article on basically talking about how long seeds are viable after saving. And it's one of the most complete that I've been able to find.
Okay. Another question about cold storage. Can any seeds be frozen to have a longer shelf life?
I think it depends on the seeds. Certainly many seeds, those that are native to places where there are frigid winters, can survive freezing. The more tropical type plants, it's probably not the preferred way. The problem with freezing is if there's sufficient water in the seed, freezing can cause ice crystals to form. And this can disrupt the internal structure of the seed. Some seeds, of course, can survive that. Actually, for example, Japanese maple, or any maple tree really, seeds, they grow very well in the Northeast, and they can sit on the ground and be covered with snow, and they're fine. But I imagine there are some tropical plants that are not used to that type of, that did not evolve in that type of an environment, whereas freezing would be detrimental. I don't have specific examples.
Right. And this is a specific question from Alton. We've got, he had a gopher problem. So the gophers actually ate the delphinium roots, but they left the tops and the pods were green at the time. He's wondering whether you think there might be viable seeds from this, or if there may be a loss.
Yeah, it's an experiment worth doing. I would save the pods, let them dry out, open them up and see what the seeds look like. If the seeds are, very poorly developed, misshapen, and don't look like they reached any type of maturity, then it's probably unlikely that they would be germination competent. But if the seeds look like you would expect mature seeds to look like, that's not a guarantee that they will germinate, but it's an indication that there's a higher probability of successful germination.
All right. And then Nick, do you happen to know how or whom decides what seeds are being stored in that world vault?
I think it's open to basically the world. Countries have specific sections in that seed vault. It's actually the site, if you simply search Svalbard Global Seed Vault, you'll go to the site. They have very interesting information there. There are some videos. It's quite a project, and it's admirable what they've undertaken, because it really is important.
And then last question I have, it was more of a mention that you didn't bring up anything about winnowing. And I'm not sure what winnowing is, but is that term familiar to you?
It is not. If we can get a definition, I can comment, but it might not be very informative.
Okay. All right, we'll give it a moment. Anybody else that has any questions, go ahead and post them now. We're caught up for the moment. Yeah. So Nick, do you recommend anything for fending off bug weevils in seeds?
I have no specific recommendation. I have no specific experience with that problem. A general, very general recommendation is make sure you eliminate moisture as much as possible. You know, seeds are fine being very, very dry in the absence of moisture. Other living things aren't necessarily. So by removing moisture, you may minimize any type of infestation. So make sure the seeds are dry. Maybe even use artificial means like silica gel packets to remove moisture.
Okay. And then we did get a definition of winnowing. So it's the process in seed saving. So it's separating the seeds from the chaff. So if you have a particularly messy seed pod, how do you go about separating the seeds from the miscellaneous organic matter?
Yeah, that is a problem with some seeds. Actually, for example, the basil seeds that I showed, you may have noticed, there's a bunch of small debris there. And for larger pieces of debris, what I like to do is I put it in a small pan, and if you gently blow across it, it will remove some of the chaff, so to speak. But frequently, the seeds also move a fair amount under even slight wind or slight air movement. So it's kind of a trade-off. You know, you want to retain as many seeds as possible, but you don't want to have so much debris that it's detrimental to the seed storage. But in most cases, it's probably not too big of a concern because, you know, just think about what happens in nature. None of that is removed. It all just goes into the ground and the seed still germinates and moves away from it.
Okay. All right. Well, that's all of our questions. I do want to thank everybody, especially for the active dialogue at the end. It's great when we receive the extra questions because there's all sorts of more information that we can gain from all of this. Okay. And then, Nick, do you have a point of contact if they do have further questions after the class? Should they be going Master Gardeners, or do you have another reference that they should use?
Yeah, you know, the best strategy for anyone that has questions is to contact our Master Gardener Helpline. This is the information here. You can basically email and it'll get to somebody. We have 3 locations that you can actually visit, although the times are somewhat limited. There's a location at Elkis Ranch, which is out in Half Moon Bay. We have a location in Redwood City at the Veteran Memorial Seed Center. And there's also a location in San Francisco botanical garden, library. And you can see the hours here, and just searching Master Gardener Helpline, and all Master Gardener locations have helplines, but if you want to be interested in the San Francisco San Mateo one, specify that, and you will get the information for contact, and the information for contact is here also.
All right. Well, and then we have quite a few thank yous for you, Nick. Just lots of compliments of how really this is one of the greatest seed lectures they've heard, and everybody's learned a lot. So
Thank you. That's
really, yeah,
that's very good to hear.
really grateful for you coming in and teaching for us, and I hope we can do this again. Yeah,
Yes.
yeah.
Well, thank all of you.
And everybody, just so you know, this class, it'll be recorded and on our Lingso website. It's lingsogarden.com. All of our clinics that we do over Zoom are recorded and posted there, so you can go back and see other classes as well. There's a lot of great information, and this class will be posted at least by Friday. It'll be uploaded to there. All right. Thank you all for joining us. All right. Have a great afternoon, Nick. I'll see you next
You.
time. All right. Bye.